first of all, I would like to thank both of our speakers for joining with us today amid their very busy schedules. So on behalf of the Nutrition Society and the department and for our students, I'm very thankful for both of you for agreeing to join with us today. So as I said, we have two very important academic scholars who are also happen to be prestigious alumni of the Department of Applied Nutrition. So I'm sure this is a really great moment for our students to look up to them, as well as I believe this is a really great moment for our speakers. It's probably a full circle for them being students and being part of the Nutrition Society and coming back after achieving so much in their academic and professional life and giving back to our student community. So I think today is one of the best days that we are having in terms of our virtual webinar series. Now, if everyone want to refresh in terms of what we are doing in the Nutrition Society, for the past couple of months, we've been arranging different, we've been inviting different scholars and academics to come and join with us and deliver virtual webinars for our student and academic community. So we are going to maintain that tradition. And today we have a jam-packed schedule. We have two speakers joining from us from different ends of the world and going to deliver two very important and timely topics related to gut health. I'm pretty sure now everyone has had a chance to go through the flyer and they know what they are going to talk. Now, as I said, we are having a busy schedule. So from 9 to 9:45, we have Dr. Thilini, who is going to speak to us about short and long-term dynamics of human gut microbiome. And then we have Dr. Madhawa, who will take right after Dr. Thilini to talk about dietary pattern networks associated with gastric cancer risk from 9:45 to 10:30. And then we are going to have a joint discussion. We will be opening the discussion floor around 10:30, and we have about half an hour to discuss any questions or to share anything that you think is important or valuable, or if you need to get more details from our speakers. So that's the general plan for today. In the meantime, if you have any questions, you can bring them to our discussion session, which we will be going to open at 10:30. In the meantime, feel free to type in and send any questions or concerns you have into the chat box, and we will divert them to our speakers at around 10:30. So now I will take just a couple of more minutes to introduce our prestigious alumni, because they definitely need an introduction. So our first speaker is Dr. Thilini. As I said, she completed her bachelor's degree in food science and nutrition from the Wayamba University. And after that, she completed her master's and postdoctoral studies, subsequently in Postgraduate Institute of Agriculture, Sri Lanka, as well as from University of Auckland. And now she's working as a postdoctoral fellow in the field of diet and periodontitis in University of Sydney. So she is an emerging medical researcher in the area of microbiome. So she received a prestigious award in 2014 from University of Auckland as a part of her doctoral fellowship. So she has been working extensively in gut microbiome research in the Liggins Institute of the University of Auckland. And she completed her PhD studies in health sciences in 2019. And since then, she has been working in the University of Sydney. So today, as I said, she's going to talk about short and long-term dynamics of human gut microbiome. And our second speaker is Dr. Madhawa Gunathilaka. He's also an alumni of our department and our faculty. So he graduated in 2015 from our faculty. And then he followed his Master of Public Health at National Cancer Center, Graduate School of Cancer Science and Policy in South Korea in 2018. And he continued his studies to postdoctoral studies and graduated 2020 from the same school, I believe. Probably not. A National Cancer Center, Graduate School of Cancer Science and Policy, South Korea. And since then, he's been working as a postdoctoral research fellow at National Cancer Center, South Korea. So please apologize if I have, missed anything, giving a brief introduction in terms of your bio, you can definitely introduce yourselves if you feel like that I have missed out anything. So I don't want to waste more time. I'm going to give this opportunity right to Dr. Thilini. Dr. Thilini, you will be able to share any slides right now because I have co-hosted you. So you can right away start and we can, yes, we can see your slides. Yes, great. So the Zoom floor is all yours. Thank you very much.
Can you see my slides now?
Yes, yes, we can.
Great. Thank you, Dr. Sushanti. And again, I'm so grateful to give this talk to you all. And thank you again for the very kind introduction. Yes, as Sushanti mentioned, I did my PhD in Liggins Institute of the University of Auckland, and I am going to present some of the studies that I completed under my PhD in today's talk under the topic of long and short-term dynamics of the human gut microbiome. So in today's talk, I'll be talking about three main projects, results from three main projects. So firstly, I'll be talking about the changes of the gut microbiome of children born very preterm compared to children born at term. Secondly, I will be talking about the long-term changes of the gut microbiome of an artist called Billy Apple from the samples we collected from 1970 and 2016. Lastly, I'll be talking about this very exciting pilot project that we completed in which we transferred gut microbial content from healthy donors to severely obese adolescents. So the gut microbiome. So as we all know, we have more microbial cells in our body compared to our own cells. So at cellular level, we have approximately 30 trillion human cells, but there are 39 trillion microbial cells living in and on our body. So these microorganisms don't do any harm to us. In fact, they protect us from becoming ill. So at the gene level, we have, you can see the vast majority of our cells are not human. So they are microbial. That's why we say we are only 43% human. So at the gene level, so we have more microbial genes than our own genes. For example, if we take the total number of genes in our body, 99% of the genes are microbial. So the microbes living in and on us and their genes contribute to unique metabolic functions in our body. So the microbiome is very important for our health and disease. So there are two terms, so microbiota and microbiome. So if you don't know the difference, I'll briefly tell you what the microbiota is. Microbiota is the microbial communities living in and on our body, and microbiome is the term which we use for the microbes living in and our body and also their gene content. So as soon as we are born, we acquire a major dose of microorganisms from our mother. So the factors like the type of delivery, whether it's C-section or vaginal birth, and the type of feeding, whether it's breastfeeding or formula feeding, and the environment, illnesses, antibiotic intake, they all shape up our microbiome. So you can see the early life microbiome development is very important for our health. So from birth to 3 years old, our microbiome is changing a lot and it becomes stable, and at the age of 3 it represents an adult-like microbiome. Sometimes, unfortunately, there can be some early life adverse events. So one such early life adverse event is preterm birth. So I'm sure you know there are different stages of different types of preterm birth. So there can be so the preterm birth is the birth less than 37 weeks of gestational age. From 37 to 32 weeks, it's an the preterm birth, and if it's less than 32 weeks, we call it very preterm birth. If it's less than 28 weeks, we call it extremely preterm birth. So this preterm birth is very common, and this preterm infants, they are treated differently compared to their term counterparts. For example, they undergo a lot of physiological stress and they are given intravenous nutrition, dietary fortification, and because these infants are more prone to get infectious diseases, they are given antibiotics and they are given steroids. So because they are treated differently, we hypothesize that they have a different microbiome. And research have shown that delayed and altered gut colonization in preterm infants. However, little is known about the microbiome development when they become older. So we, in our research project, we have asked the research question whether these children have different microbiome composition, microbiota composition compared to their term counterparts. So to answer our research question, we recruited 5 to 11 years old healthy prepubertal children. So in that cohort, we had very preterm children, 51 very preterm children and 50 term children. And then we collected their medical history from the mothers and also from children. For example, maternal age at childbirth, pre-pregnancy BMI, delivery mode, type of feeding, etc. And also we collected demographic data such as ethnicity, anthropometric data, clinical data, insulin and glucose measurements, and dietary data. And also we collected stool samples for microbial analysis and also for the metabolomic analysis. So as I mentioned before, we collected stool to tubes containing RNA later, and they were brought to Liggins Institute where I extracted DNA and RNA from these stool samples to sequence and see what the microbiome looks like. And this slide summarizes the stool and plasma analysis. So as I mentioned before, we had 101 children in our cohort, and out of 101, 91 children provided stool samples. And then from these stool samples, I performed 16 is rRNA gene amplicon sequencing. So I'm not sure whether you know what this technique is exactly, but the 16 is rRNA gene is the unique, it's a universal gene to bacteria, and it's like an identity card. We can sequence the sequence 16 is rRNA gene and see what bacteria are present in these samples. And then from these 91 children, we had a subgroup of 41 children. Because this other sequencing techniques are really expensive, so we actually had to limit the number. So in that from that 41 samples, I performed whole metagenomic sequencing. So whole metagenomic sequencing, we can see the microbial functions and the microbial compositions in those samples. And also I performed metatranscriptomic sequencing. So that's RNA sequencing to see what are the active microbes in these samples and what are the active functions in these samples. And we also performed fecal and plasma volatiles from solid phase microextraction and gas chromatography mass spectrometry. And we also analyzed fecal and plasma amino acids from ultra performance liquid chromatography. And also fecal calprotectin was measured using ELISA. So this slide summarizes the demographic and demographic characteristics of this population. So as you would expect, gestational age of very preterm children was less compared to term children. And from the other anthropometric characteristics, we could see these preterm children, they were younger and they were leaner compared to their term counterparts. And as I mentioned before, we analyzed the microbiome of these children from 16 rRNA gene sequencing. So this graph summarizes the microbial composition of preterm and term children. And on the bottom, you can see on my left the microbial composition given as genus of preterm children, and these are the microbial composition at genus level of term children. So there's no specific pattern between preterm and term children. So yes, from these results we could see that there's no difference in the microbial composition from 16S rRNA gene sequencing between preterm and term children. Then we looked at some other microbial parameters. So one such parameter is alpha diversity. So alpha diversity is showing us within the sample microbial diversity. So there's no difference between preterm and term. For example, this first row, these um, four graphs are actually a microbial composition, the alpha diversity measurements between preterm and term at species level. Second row is at genus level, and third row is at family level. So at each level we didn't see any difference between preterm and term in any of the alpha diversity measures. So there are four different alpha diversity indices here, like Chow1, observed species, Shannon index, Simpson index. We couldn't see any difference between groups. And then we looked at between sample diversity using beta diversity measured. So again, there's no difference between term and preterm. So simply if there are two different clusters, you see that there is a difference between groups, but here there's no different clusters. They are all everywhere. For example, these blue dots represent the microbiome of term children, and the red dot represent the microbiome of preterm children. And there's no difference. And we also look at the beta diversity between other co-founding factors, for example, male versus female, and between the different ethnic groups, and the type of delivery, C-section versus vaginal, and antibiotic intake, whether they had antibiotics or not, and the type of feeding. And again, there's no difference between these groups, even though the sample number is less when we subgroup them. So as I mentioned before, these co-founding factors like sex, ethnicity, mode of delivery, intake of antibiotics, mode of feeding, they didn't show any impact on bacterial composition. Then, as I mentioned before, we also looked at the whole metagenomic sequencing data in the subgroup of 41. Again, there's no difference in microbial taxonomic composition or functional potential. So I'm only showing you the taxonomic data. So this graph shows the relative abundance of top 20 most abundant bacterial genera. So on my left, it's the genera in preterm group. On my right, it's term microbial composition at genus level in term group. So simply there's no specific pattern between these two graphs. And we also looked at the significance using LDA score, linear discriminative analysis score, and there's no significant difference. So interestingly, when we look at the bacteriophages richness, we did see a difference. So what are these bacteriophages? So bacteriophages are viruses that attack bacteria. So these viruses, these bacteriophages are unique to bacteria, and it has been shown that these bacteriophages have an association, they have an impact in the immunity in our human body. So interestingly, when we look at the bacteriophages richness in preterm group and term group, preterm group had less bacterial richness compared to term group. And we also looked at the fecal and plasma metabolite profiles in preterm and term children. So especially this arginine in plasma of preterm children. So here the plasma arginine in preterm children is significantly low compared to term children. But when we look at the fecal arginine levels between term and preterm children, we didn't see any significant difference. However, Fecal arginine is more variable in preterm than term children. So the majority of preterm arginine is less than 100 micromolar per liter. Then we also looked at the fecal branch chain amino acids and hydroxyproline. So I'm sure you know the different types of amino acids. So branch chain amino acids such as valine, leucine, isoleucine, hydroxyproline. They are hydroxyproline is not a branch chain amino acid, but it is a type of amino acid. But they are lower in preterm children compared to term children. And we also looked at the calprotectin. So calprotectin is a marker of inflammation. So when we looked at the fecal calprotectin, we could see that preterm children had higher fecal calprotectin level compared to term children. So let's move to the discussion of this first part. So as I mentioned before, there's no difference in bacterial profile, and that is consistent with other studies. So microbiome is resistant to early life stresses. So evidence linking the bacteriophage population structure to the preterm phenotype and ability to adapt to environmental change remains uncertain. So, however, we postulate that the reduction in phage community richness in preterm children that we observed was due to the developmental differences in the microbial population in the neonatal preterm environment. So there's another interesting finding from previous studies. The children born very preterm have a reduced capacity to synthesize arginine because they have a limited expression of arginine biosynthetic genes. So we also observed in our results there's a decreased plasma arginine levels in these preterm children. So that reflects the long-term reduction in host arginine biosynthesis or altered bacterial synthesis. And as I mentioned before, these preterm children had higher calprotectin levels. So that's consistent with impaired gut epithelial integrity, reducing the selectivity of transfer of gut microbial metabolites into the bloodstream. So I didn't show you the results, but we actually looked at the insulin resistance of these children, these preterm and term children. And preterm children had higher insulin resistance compared to term children. So insulin resistance is associated with the inflammation. So this higher calprotectin level in preterm reflects that too. So based on our results, we conclude that while potential early differences in bacterial microbiota were lost over time, we observed reduced phage richness even after 5 to 11 years in children born very preterm. And these changes may impact on the ability of preterm children's microbiota to respond to acute environmental changes. So, however, we need longitudinal studies of microbiome and child development, and these studies should incorporate bacteriophages analysis because that's not something which is very common in current microbiome studies. So this is the publication we completed few years ago, I think last year. So if you are interested in reading this paper, you can check it out, and it has all this information and some other additional results which I didn't present today. And let's move to the second part of my presentation. So in the second part, I will be talking about the comparison of gut microbiome 46 years apart. So as you would imagine, there's lack of long-term longitudinal studies which investigate the stability of the microbiome. However, we were lucky that we found this artist called Billy Apple. Unfortunately, he passed away early this year. So he had kept his stool samples containing toilet tissues. When we met him in 2016, he used those samples, these tissues, for his one of the art exhibitions. So that's a different story. But yeah, so it was interesting. We found these samples, and we wanted to characterize the microbial composition. And what we did, he already had this toilet tissues containing stool from 1970, and we collected another set of samples, another set of tubes from different places, and you can see from this bar graph, the first three bars shows the bacterial composition from 1970 samples, and the last three bars shows the bacterial composition from 2016. So the main difference is you can see this brown color um, sections. So you can compare between 1970 and 2016 the bacteroids, genus level data. So genus bacteroid actually reduced from 1970 to 2016, and also Faecalibacterium levels have been reduced. And the other interesting thing is if you see these purple color ones, there's very less amount in 1970, which actually present Prevotella, but 2016 you can see there's a huge increase in Prevotella in those samples. So Prevotella, we don't know what are the actual species because this data is from 16S rRNA gene sequencing, but some of the Prevotella species are associated with inflammation, systemic inflammation, increase in some of the cytokines like interleukins 1, 6. And then we also did another analysis. Again, I'm not sure whether you are familiar with this human microbiome project, but in human microbiome project in phase 1, they characterized the microbiome of healthy individuals from I think it's 300 healthy individuals. They took samples from different places, skin, from the oral cavity, stool samples, urogenital tract. So they characterized microbiome of different places. And this data is from gut microbiome analysis. And this blue dots represent microbiome of healthy individuals. So you can see I actually compared Billy's 1970 samples and 2016 samples with human microbiome healthy subjects data. So you can see this red dot represents Billy's 1970 microbiome. It's actually clustering with healthy individuals microbiome. So Billy was healthy when he was, I think, 36 years old in 1970. And but you can see Billy's 2016 samples when like after 46 years, it's actually deviating from healthy microbiome. So as you would imagine, with the age, his microbiome has changed. He had lived in different places in the world, and at that time he was having some health problems. He was under certain medication. So because of all that, we can see that there's a shift in microbiome from early age to late ages. And then we looked at the level of conservation, and this particular group, faith, they actually looked at the diversity of microbial population with the time. So they collected samples within 5 years, and they actually extrapolated that to 10 years. And you can see the diversity of microbial population with the time is reducing and it becomes stable. And what we did, we extrapolated this graph up to 50 years, and Billy's 46-year-old sample lies here. So what it says, like 45% of Billy's microbiome is stable from throughout these 46 years, which is amazing because as I mentioned, he had lived in different places, he has been aged and under a lot of medication, but still some part of microbiome is stable with the age. So from this, again, in addition to that, I didn't show you, but we found some of Billy's genomes, gene alleles are actually associated with some of these species. So what we conclude from this study was that the core part of our bacterial population remains stable as we age, and that at least some of the bacteria are actively selected by our genes. So again, we published this study in Human Microbiome Journal, and if you are interested in, you can check that out too. So with that, I will be moving on to the last project that I am going to talk about today. Um, so that is about the fecal microbiome transplantation from healthy donors to severely obese adolescents. So today I'm going to present to you the results from a pilot project. So the basis of this study is this mice study. So in this mice study, what they have done, they have taken the microbiota from obese twin to lean recipient mice, and these recipient mice were on low fat high fiber diet for certain period of time, and then they checked the adiposity. These mice have developed adiposity, they had increased adiposity. So the other group, so they have taken the lean twin, and they took the microbiota from this lean twin and transplanted into again the recipient mice, and they were on low fat, high fiber diet. However, they didn't develop adiposity, they stay lean. So this is very interesting. So what we wanted to do, we wanted to take a stool from healthy donors and to transplant them in severely obese adolescents and see whether they are going to reduce their fat mass, or whether they're going to reduce their some of the cardiometabolic parameters. So first of all, we had to find the donor. So that was a very difficult part, because we had to go through this very rigorous procedure to find these donors. We call them super donors. So they were free from all the diseases. They should have normal fat percentage, should be in normal BMI range, shouldn't have any other disease condition. They shouldn't be taking any prebiotics or probiotics, so they had to undergo a lot of tests. So after we did clinical and safety evaluation, I collected their stool and mixed it with saline and made these small capsules. We call them fecal capsules. Then I had to check these capsules for viability and diversity check. So there was the small part of experiment that I had to complete. I'm not going to show the results, but from that experiment, we found out that we can keep these stool capsules at -80 for 6 weeks without actually changing their microbiome, because we wanted to transfer the live microbiome from donors to recipients. However, at the time of collection, 30% of the microbes are dead. That's we couldn't avoid that. So after we checked that, we recruited recipients. So we were aiming for 12, but finally we had only 11 recipients. So they were severely obese recipients. So we had to assess them for eligibility, whether they had some other diseases or whether they are on medications, antibiotic, prebiotic, like supplements. And after baseline assessments, these recipients were given bowel cleansing. So after they are given bowel cleansing, they'll flush all their bowel content. And after that, we randomize them into two different groups. So one group gets the fecal transplantation, so fecal capsules. The other group gets the saline capsule, so that's a control. And then we followed them up for 6 months. So at baseline, we collected data, and 6 weeks, 12 weeks, 26 weeks, we collected their stool samples to check whether there are any differences in the microbiome. And also we collected some other parameters like glucose level, insulin resistance were calculated, and the blood pressure and lipid profile was analyzed. So So as I mentioned before, this is a pilot project and the sample size is very low. However, we looked at the baseline characteristics between controls and the FMT group. So FMT stands for Fecal Microbiome Transplantation group. So except a few parameters like total cholesterol and LDL cholesterol, LDL levels, the other factors were not different between groups. For example, age, BMI, C-reactive protein, insulin resistance, so total body fat percentage, they were not different between these two groups. So let's look at the results from this project. So again, this I will walk you through these results. So these dots, for example, this black dot represent the microbiome of donors, one of the donors. And these two samples are from the same donor, donor number 1, and represent the microbiome of donor 1. And donor 2 here, 3, this purple dot, and here donor 4, and yeah, so we had only 4 donors. So here I'm showing you the microbiome shift from baseline to 6 weeks. So first of all, to start with, this F1 is the person who had the fecal microbiome transplantation. So this dot represent the microbiome at the baseline. So you can see this arrow represent the shift. So there's a huge shift from baseline to 6 weeks. So it's same with F2. There's a shift, and it's same for F3. If you look at F3, F4, F5, yes, you see the shift from baseline to 6 weeks. And then I mentioned before, we followed them up for 26 weeks. So when we looked at the 26th week samples, we still see that shift. So there's a shift from baseline to 26 weeks. And the other interesting thing is it's actually shifting towards one of the donors, not all other donors, like donor number 1. So that's very interesting because we actually mixed these donor samples when we made these capsules, but donor 1 sample is dominating because this person had the highest diversity in their microbiome, which is very interesting to see. And then I mentioned before, we had this control group, the saline group. And again, you see these donor samples are in this group, and the saline group, like say saline 1, so the recipient who had the saline capsules, actually didn't shift the microbiome from baseline to 6 weeks at all. So it's same except these two, these three, like there's a little shift, but compared to FMT group, it's actually very, very low, and some there's no shift at all. And we also looked at the microbiome at 26 weeks in this saline group, and actually circle them to see how their shift is. For example, in the saline 1 recipient, this dot represents the microbiome at baseline level, then 6 weeks, 12 weeks, 26 weeks, and they are actually clustering together, and there's no dominant shift. And it's same with saline recipient 2, 3, 4, 5, 6. There's no shift at all, and it's actually the shift is not they don't have any specific direction like we saw in FMT group. Yes, so from this is a pilot project, and from the pilot project, we concluded that yes, this fecal transplantation is safe and there were no side effects. And the fecal microbiome transplantation is capable of shifting the recipient microbiota successfully, and that engraftment of microbes in the gut of recipients lasted for a minimum of 26 weeks. Yeah, so that was the finding from the pilot project, and we did made a documentary from this study, and then we actually continued with the main trial, in which we had 2 groups, female and male, and again the same methods were followed. And unfortunately, we didn't see any even in pilot project or the main trial, we didn't see any change in the fat mass or any other cardiometabolic parameters. So we published so many papers from this particular study, and you can check these if you are interested in. And so I want to, this is not just my work, we had so many different people working on these projects, and I worked with different people from metabolomic field, microbiome field, bioinformatics, and I want to thank all of them, and especially all these subjects who provided samples for our analysis. So moving on, just want to briefly tell you what I am currently working on. So I was working with gut microbiota, and now I'm working with oral and gut microbiome. So our theme in current research is the oral health and the general health. So there is a link between oral health and general health. So in our oral cavity, there are a lot of different microbes, and they produce a lot of different metabolites, and they are transferred to systemic circulation, and some of the metabolites and some of the microbes actually have a very adverse impact on the systemic inflammation, which actually affect different which actually links with different disease conditions like diabetes, CVD. For example, I'm currently working on a different project in which we look at the gut microbiome and the oral microbiome of the diabetic patients, and also we are characterizing the microbiome of the wound samples, the foot ulcers of these diabetic patients. And also there is a link between CVD and gut microbiome and also oral microbiome. So this current project in which we look at the gut and oral microbiome of CVD patients. And there are a lot of research around the area of anxiety and depression, and there's a well-known link between gut microbiome and anxiety, depression, autism. And what we are currently looking at is the oral microbiome, how oral microbiome is linked with these conditions. And we are running some mice experiments as well. There are 2 mice projects. So in one, I looked at the effect of diets on periodontitis. So periodontitis is a gum disease. There are previous research linking this gum disease and gut microbiome. So we actually wanted to look at the diet effect on periodontitis and diet effect on the gut in this periodontitis mouse model. And there's another research on the fiber intake and periodontitis. So the fiber, what is the effect of fiber on periodontitis, and what's the effect of fiber in oral and gut microbiome? And lastly, I'm working on this clinical trial, so in which we look at the pregnant women who had early miscarriages and their microbiome, gut, oral, vaginal microbiome. So these are the current projects that I'm working on. So with that, I would like to conclude my presentation. So thank you very much for listening, and I'm happy to take questions when it's time to take questions. Thank you.
Thank you very much, Dr. Thilini Jayasinghe, for that very informative presentation. I'm sure it was an eye-opener for all our students. And if you have any questions and clarifications, I'm sure she will be glad to answer your questions and concerns at the discussion time. Now, we are right on time, and we are moving to our next presenter. Our next speaker is Dr. Madhawa Gunathilaka, and he's also going to talk about the gut health. So let's hear it for Dr. Madhawa. Dr. Madhawa, you should be able to share your slides now. Yes.
Thank you, Dr. Tushanti, for your very kind introduction. And it is my great honor to have this opportunity to have a talk with today. And thank you for giving the opportunity by the our Nutrition Society. So, and thank you, Dr. Thilini, also for your very interesting talk. And and so can you see my slides clearly?
Yes, we can see your slides. You can just click on the slideshow. Yes.
Yes. So my talk is today about the dietary pattern networks associated with gastric cancer risk. And so the contents of my presentations are mainly three parts. First, I will introduce the definition and some what are the dietary patterns, and then what are the methods of the identification of dietary patterns. So basically, I will consider three methods: the Gaussian graphical models, GGM, principal component analysis, and reduced rank regression. So, and then in my third section, I will introduce the application of the dietary pattern analysis using Gaussian graphical model in the association of the gastric cancer risk that I was able to work in during my PhD studies. So for the definition of the dietary patterns, the dietary patterns reflects the whole foods or the combination of the consumption, temporal distribution of intake, and their habitual pattern. For instance, like we can consider like snacking and certain food preparation methods. So those things could be considered. For the definition of a dietary patterns, it can be the quantities or the proportions, a variety or combination of different foods and nutrients in diets and the frequency with which they are habitually consumed. So when it comes to the diet patterns and the cancer risk, so this research area has a very rapid expansion, specifically when it comes to dietary patterns and the cancer association to make the cancer prevention guidelines by considering various dietary factors. For instance, the popularity of this dietary patterns research is reflected in the kind of a very updated research report based on the previous evidences, which is published by the World Cancer Research Fund, American Institute for Cancer Research, third expert report published in 2018. So they have given a considerable space to address the dietary patterns and how the cancers are associated with the dietary patterns. And multiple dietary patterns and the indexes have been developed previously that reflects the dietary guidelines. So here I have listed certain indexes or the patterns, namely like Healthy Eating Index, World Health Organization Health Diet Indicator, New Nordic Diet, Chinese Food Pagoda, Mediterranean diet, and dietary approaches to stop hypertension. Most importantly, for the characterization of the dietary patterns or the indexes, we have to have a good understanding or the knowledge of the foods or the beverages or nutrient intake or the combination of those factors. And in the epidemiological studies in the dietary patterns and the cancer, the kind of as a nutrition assessment tool, this self-reported food frequency questionnaire has been used to assess the usual diet of the study participants. And normally the typical dietary pattern analysis can be divided into major two groups based on whether how the patterns are determined, namely the empirically or also known as posteriori approach, or otherwise using an investigator defined method called priori approach. So this chart shows the dietary pattern analysis method, and the investigator-defined method is kind of a hypothesis-oriented method. And in this approach, we have to consider about the previous existing knowledge of the diet or the food groups. And the certain examples are the selective diets and indexes or scores. In this case, we do not need to consider about the dietary intake data of a study population. When it comes to the exploratory approach, it's a data-driven approach that we can have to use certain statistical approaches to derive the dietary patterns. So in the nutritional epidemiology research, there are several approaches. So I have listed three here, the GGMs, PCA, and RRR. So the PCA and RRR are kind of and factor analysis. So those are kind of traditional approaches that can be used in the nutritional epidemiology research to derive dietary patterns. And when it comes to the Gaussian graphical model, it's a novel approach to application in the nutritional epidemiology research to derive dietary pattern. Actually, this model has been applied in the genomics and the metabolomics data to identify their internal pattern. However, when it comes to the dietary data also is multidimensional. So we can apply this approach to derive the dietary pattern. So in my presentation, I will mainly focus on the GGM. So this diagram shows the certain dietary patterns derived by the posteriori approach, which are associated with the several cancer types. And mainly the two patterns can be posteriori identified, like either healthy pattern or also known as traditional or a prudent pattern. And the other one is the unhealthy pattern, also known as Western pattern. So several type of cancers including GI cancers, are associated with those kind of healthy and unhealthy dietary patterns here. So those are listed, and also these evidences are mainly based on the cohort studies and also the case control findings. And then this figure shows the dietary patterns derived by the priori method, which are based on the previous existing knowledge of the dietary information. So in this case, we can see like healthy eating index, Mediterranean dietary score, and those kind of thing. And specifically when it comes to gastric cancer, the Mediterranean dietary score is associated with the decreasing risk of gastric cancer. So I'm not going to talk about the priori approach in my presentation from this onward. So in the second part of my presentation is the methods of identifying dietary patterns. So as we all know that we normally do not eat a single food item or a single nutrient at the one time. So in our diet has a complexity in nature, and they are very highly correlated between intakes of various foods and the nutrients. And it makes difficult to examine their separate effects. So that we have to consider about the comprehensive dietary variables that allow for the intake of many foods that may show a greater effect on the disease than a single component. And that kind of comprehensive variable can reflect the specific dietary pattern, and that represents a more accurate picture of the diet rather considering the isolated food. So going more in deep, the pairwise correlations between food groups can be more informative, and if they are independent of the effect of the rest of the other food groups. In order to get the dependency of the various food groups in the dietary data, so the pairwise correlations is very informative to assess the correlation between food groups that control for the other food groups in the underlying data. And this may be important in understanding how foods are consumed in relation to each other. So the existing methods of the deriving dietary pattern analysis like TCA, so those methods may require certain subjective choices during the data analysis. However, when it comes to the innovative methods like GGM that provides additional information that might be advantageous over the conventional approaches. And they will provide improve our understanding of the complexity of eating behaviors, and also to overcome the limitations associated with the traditional methods of dietary pattern analysis. So then I will introduce the GGM, and these are graphical methods that identify the conditional independence structure in the data set by assessing the pairwise correlation between two variables by controlling for the rest of the other variables. And this assumes the multivariate normal distribution for the underlying data. And also it infers a direct relation between variables in a given data set without considering the prior knowledge. Especially when it comes to the pairwise correlation between two food groups, controlling for the others food groups, it can identify two main parts. The first one is the internal structures or the patterns in the underlying data set or the topology of that data set, and then the relation between either direct or indirect relation between the food groups consumed in the, identified network. And these networks also identify the key interrelated food groups that may be potential candidates for further investigation into the confounding structure to observe when observing the association with the disease risk. And this leads to a good understanding of the biological relations between diet and health status. So when it comes to the theoretical background of the Gaussian GGM, so they are probabilistic graphs that can be used to analyze and visualize the dependence or the interdependence or the partial correlation coefficients between the variables. The graphs in this method, there are graphs, and so those graphs with the set of nodes and edges that represents the conditional dependency relation between the food group variables. And if there is a missing edge between two variables, that represents the conditional independence between the variables given rest of the other variables. And so this is called pairwise Markov property, and it is quantified in terms of partial correlation. So this is the way of representing the Gaussian distribution. And as you can see here, this is the data, X is the data matrix with the n number of observations and the p number of variables with the mean vector mu and the sigma covariance matrix. So this is Gaussian distribution. So from the inverse of this covariance matrix, we can obtain the conditional independence of two random variables after controlling for the rest of the other variables. So the inverse of the covariance matrix is also known as the precision matrix. And suppose that, let's see, there is a two random variables called P1 and P2, and they are connected with the edge, which is the correlation here. So the edge corresponding to the non-zero elements of this precision matrix. And the estimation of the conditional independence in the precision matrix or the inverse of the covariance matrix forms the basis of the Gaussian graphical model. However, when we work in the real world data, which are very highly dimensional and multivariate with normal distributed data, it's very difficult to have this kind of clear uh, graph to represent. So normally the precision matrix does not contain zero values or a few zero values. So that means this graph is very highly concentrated and each nodes are connected to each other nodes. So very difficult to identify the internal patterns or the topology structure of this method. So in that case, we have to think about a kind of relaxation techniques of this graph, which is of course known as making a sparsity assumption. So that is the place where we require adopting a regularization technique that enforces the sparsity to the precision matrix. So this mathematical representation is for that sparsity based on a regression technique called Lasso. And so based on this assumption, we can make the precision matrix to sparse, and then we can obtain the graphical representation with a clear and easily identified pattern. So as I mentioned, the regularization technique is applied based on the L1 regularization, which is known as Lasso regression. And so this is where the simple linear regression function is given here. And this Lasso adds the absolute value of the magnitude of the coefficient, which is beta, to the penalty term as a penalty term to the loss function. So here is the penalty term, and this addition of this penalty term can shrink the less important features coefficients to 0, so that we can remove the less important features by retaining the most important features in the underlying data set that will be utilized for the further analysis. So in here, very important thing that we have to consider is the lambda, which is known as the high regularization parameter. So obtaining the regularization parameter is critical in the GGM analysis, so that we can identify a clear pattern of the underlying data. So in order to simplify this technique, so I have shown here the practical way of doing this method using two R packages called Graphical, Lasso, and the huge packages. So after obtaining the matrix or the data matrix or the covariance matrix from the underlying dietary data, so using the Graphical, Lasso packages, we can obtain the inverse of the covariance matrix and series of lambda values like in a decreasing order, and then we can select the optimum lambda value using the huge package. So series of lambda values will be obtained, and then the corresponding precision matrices also will be obtained. So after using the huge package, and then we can obtain the optimum lambda, so that the corresponding precision matrix that is related to the optimum lambda value will be utilized for further visualization and the analysis. So, and then after obtaining the optimum lambda value, so we can, using QGraph packages, we can visualize this precision matrix as a graphical way. So that is the basis of the Gaussian graphical model. So in my presentation, I will show you in the application how it can apply. And for the principal component analysis, it's kind of a dimensionality reduction technique to reduce the dimensionality of a data set consisting of a large number of interrelated variables while retaining as much as possible of the variation present in a data set. And this can be achieved by the transforming to a new set of variables called principal components. And so from the underlying data variables, we are going to reduce the dimension to a selected principal components called variables called principal components. And those components could be used as an independent variable. So those should be uncorrelated to each other. And also the first few components can retain most of the variation present in all of the original variables. So the other important thing is the component loadings or the factor loadings in this analysis. So those are the correlation between the original variables, which are X, and the components, which are newly derived variables, which are Z. So we can find the component or the factor loadings in this method. And the components can also be rotated to simplify the structure of the loadings matrix and for interpretation of the results. And the other important thing that we have to consider the component retention for this PCA analysis. So in this case, we have to consider about the tree plot of the eigenvalue should be examined. So this diagram, this figure shows that tree plot to determine whether there's a break in the pilot plot with the remaining components explaining considerable less variation. So when you see the scree plot here, so the principal component is in the x-axis, whereas the eigenvalues are given in the y-axis. So the how many number of components should be retained has to be considered cautiously. So for this approach, there is a rule called Kaiser Rule, which is the rule of thumb. It uses the number 1. The components or the which are greater than number 1, the that eigenvalue which will be retained. However, this is still depends on the researcher and also the previous literature evidences. So the certain investigators can identify by themselves how many number of components should be retained for the as to consider as the independent variable for the further analysis. And so this shows that explain variation. So this shows the explained variation with the principal component, and this shows the cumulative explained variation. So component rotation also has to be considered in PCA. So this component rotation allows the components to be easily interpreted as the clusters of the variables that are highly correlated with the particular component. And we use the orthogonal rotation or a Varimax rotation. And this rotation technique preserves the perpendicularity of the axis. So with the application of this Varimax rotation, so the newly derived components will be uncorrelated each other. So that is a very important point. And this aiming to maximize the squared loadings variance across the variables, sum over all factors. So the third approach is the reduced rank regression, and this is a statistical method that determines the linear functions of the predictors. Predictors in a sense that the food group variables by maximizing the explained variation in the responses, or in a sense that it's a disease-related nutrient. So the important feature in the reduced rank regression is the identification of the responses. So for the definition of the responses, we have to think about the previous knowledge of the data, existing data based on the meta-analysis or a cohort studies or something like that. So, and then we can do this analysis. So since it uses both information sources like data from the study itself and also prior information, still this method is a posteriori approach method. So the theoretical background is let's say the X1 to Xn are the predictors, which are the intake of the food groups, and Y1 to Y1M, so those are the responses. So these are the intake of nutrients in grams per day or nutrient ratios. And these are the key feature of the RRR that extract the factors that explain as much response variation as possible, not the predictors like PCA. So the coefficient vectors of the extracted linear functions and the eigenvectors of the covariance matrix, and these RR starts from the covariance matrix of the responses, but not the predictors. So, and then comparison between the Gaussian graphical models and which has more strength than the PCA and the RRR. So the Gaussian graphical model observe the pairwise correlation or the conditional independence, and that helps to understand how foods are consumed in relation to each other. And it minimizes the subjective choices during the analysis, and it's easy to interpret the results, and also a ability to distinguish between direct and indirect associations between food groups consumed without considering the prior knowledge. And it introduces the sparsity, so it can select the most important key feature variables in the final model. And it leads to a much better understanding of the biological relations between diet and health status. And when it comes to PCA and RRR, so they cannot completely understand the pairwise association like GGM did, and so that cannot control the indirect effect of the other food group. And those methods require several subjective choices during the data analysis. And like for an example, for the PCA, the determination of the number of components should be retained. So it depends on the investigator sometimes. And for the RRR, like we have to consider about the previous existing knowledge of the diet to define the responses. So it needs some other subjective choices, and this do not introduce any sparsity technique also. And it's difficult to better understand the biological relations like the Gaussian graphical model. So in my third part, I will show the application of the pattern analysis using GGM, and then observe the association with the gastric cancer risk. So I applied the GGM in a case control study. So for this study, the 500 gastric cancer cases agreed to participate initially, and 1,227 healthy controls agreed to participate. So from them, they are from the participants, based on the demographic information and the dietary data availability, certain exclusion criteria were applied, and after that, the cases and the controls were matched 1:2 ratio, frequency matched 1:2 ratio, based on the distribution of age within 5 years and the sex. So after that, the 415 gastric cancer cases and 830 healthy controls were selected for the final analysis. So as a data collection tools, the structured questionnaire was used to collect the possible confounding variables like age, education, smoking, and alcohol drinking, regular exercise, and those kind of information, and family history of cancer, and validated semi-quantitative food frequency questionnaire, which has 106 food items, was used to collect their dietary intake data. So this table shows the demographic variable, the comparison of the demographic variables between gastric cancer cases and the healthy controls. So those red color indicated variables are the variables that show a significant difference between gastric cancer cases and healthy controls. Specifically, higher proportion of the cases are current smokers, and high proportion of the cases have the significantly high proportion of cases have family history of gastric cancer, and a significantly lower proportion of cases do not engage with the regular exercise and other like education, their occupation, so they are significantly different, and significantly higher proportion of the cases are H. pylori positive status. And total energy intake comparison, there's a significant higher total energy intake in the gastric cancer cases compared to the healthy control. So this table shows the comparison of the selected food groups, 33 food groups, between healthy controls and the gastric cancer cases. Actually, these 33 food groups were derived from the original food frequency questionnaire. The 106 food items were collapsed based on their culinary usage and the nutrient profile to 33 food groups for the analysis of the pattern. So this is how it looks like, as I mentioned you. So the identification of the lambda, which is the the regularization parameter, so it's critical. So in our case, it's the range is starting from 0.79 to 0.08. So this is how the graphs looks like. So for the higher the lambda means higher the sparsity. So in this case, we are very relaxed. The graph is very relaxed. So the in relation between the food groups variables are we cannot identify the interrelationships between the food group variables. So we cannot use this kind of graph for the pattern identification. So progressively, when we decrease the lambda value, so that we can see how the relationship or the correlation between food groups are visual appearing. So in our data, we identified the optimum lambda value as 0.37, and this is the precision matrix corresponding to that optimum lambda, and then visualized as a network. So these are the other graphical presentations for the other rest of the lambda values. So this is the lowest lambda, which has the very lowest sparsity and very highly concentrated graph that cannot use to easily identify the dietary patterns in this data. So there's no meaning of using this kind of graph to present the dietary pattern. So that is the reason identifying the optimum lambda and this graph. So then after identifying those that specific precision matrix to the optimum lambda value, I imported this precision matrix to YED graph editor to visualize the dietary patterns. So I identified this is for the total study population, and there are 5 dietary patterns networks were identified. The main network was composed with vegetables and the seafood. and so it's known as vegetables and seafood pattern. And mainly 10 food groups were clustered together, and in the mainly those food groups were clustered around the light colored vegetables, green yellow vegetables, and condiments and seasoning, those food groups. And this value represents the partial correlation coefficient. So the correlation between two food groups after controlling for the rest of the other food groups. That is how we can interpret this correlation coefficient value. And the other subnetworks are the snacks and the fat pattern, and then the meat pattern, and milk, dairy pattern, and the fruit pattern. So when it comes to the fruit pattern, the fruit and food products are very highly correlated. So this is for the, I derived the sex-specific dietary patterns also. So this is for the male population. For the male population, the main networks also composed with vegetables and the seafood. And the other three subnetworks were snacks and fat pattern, meat pattern, and fruit pattern. And for the female population, still the main network was clustered with vegetables and the seafood food groups. So it was named as vegetables and seafood pattern, and snacks and fat pattern, and the meat pattern, and dairy pattern, and fruit pattern. So from this, after obtaining this dietary pattern networks, there is a research gap that how we are going to convert this network into a pattern score. So addressing that research gap, I considered the network analysis or the centrality theory of the network analysis to calculate the strength value of this each node, and then combine with the log transform value of those intake variables and get their summation to calculate the pattern specific score. So that pattern specific score was used as the kind of continuous variable to observe the association with the gastric cancer risk in further analysis. So so this table shows the association between dietary patterns derived by GGM and the gastric cancer risk for the total population. And interestingly, so for the total population, so I divided the dietary pattern network score into tertiles based on the distribution of the healthy controls, and those subjects who are in the third tertile of the vegetables and the seafood pattern score showed a significantly reducing risk of gastric cancer. Interestingly, for the fruit pattern network, so those who are in the highest third tertile of the fruit pattern showed a significantly reduced risk of gastric cancer in total population. And for the male population, I identified those males who are in the third tertile in the vegetables and the seafood pattern showed a significantly reduced risk of gastric cancer. But when it comes to fruit pattern, it didn't identify a significant association. And for the female population, actually, I was unable to find a significant association. Only in the fruit pattern, the second tile showed a significant reduce, but the PP4 trend does not show any significant relationship. So, and then, yeah, so the summary of my presentation, the diet patterns reflects the habitual exposure or that represent the complexity nature of our diet or the food and nutrients, and serve as an optimal method to assess the diet and the disease relationship. Specifically, the Gaussian graphical models are the powerful exploratory data methods to derive the patterns. And the key feature is the conditional independence networks that provide a key insight to identify the consumption pattern. And the identified networks still further can be transformed to intake pattern scores and for the association with the health status. So it has a research gap in this topic still needs to be considered. So for the PCA and RRR, they are able to identify the patterns, but those methods need certain subjective choices during the analysis. according to my findings, the GGM identified the vegetable and the seafood network. It was associated with reduced risk of gastric cancer for the total population and the male population. And for the fruit pattern network was significantly associated with reducing the risk of gastric cancer for the whole study population. And so that I would like to acknowledge for my advisor, Professor Johnson Kim, for giving the opportunity to work with this interesting topic, and all the funding sources and the research team members in my lab, and also Nutrition Society for giving this opportunity to have this talk today. So thank you for your attention, and I'm happy to answer for the questions.
Thank you very much, Dr. Thilini and Dr. Madhawa, for delivering us two very important areas related to GI. And I think most of the participants here being nutrition undergrads, you two made ourselves motivated. So Dr. Thilini talked about the long-term and short-term dynamics of the human gut microbiome. We listened how the gut microbiome differs in between term children and preterm children. More interestingly, how the fecal and plasma metabolite profiles differ in preterm and term children. And also the long-term changes of the gut microbiome, 46 years apart. Finally, we listened to gut microbial transfer, which happens between healthy donors and obese adolescents. And then Dr. Madhawa talked about the dietary pattern networks associated with gastric cancer risk, which is a timely topic with very important information, including Gaussian graphical models. So now it's time to open up the discussion session. We warmly welcome all our participants to raise any question related to topics discussed today. You can raise your hand and unmute yourself to ask the question, or else you people can direct the question through the chat box.
Can I ask question from Dr. Thilini?
Yes, sir.
Yeah, so thank you very much for the interesting topic related to microbiome. So I'm wondering about the what kind of bioinformatics software you are using for this analysis?
Yeah, so we used for the metatranscriptomic and metagenome. I didn't present metatranscriptomic results today, but I did analyze metatranscriptomic sequencing data. For whole metagenomic sequencing, I used Metaflan. I'm sure you are familiar with it, right?
Yes.
Yeah, Metaflan from Hutton Howell Lab
Yes.
in Oxford.
Yes.
I'm sorry, Harvard Uni. Yeah, so they have a lot of different pipelines which are designed for microbiome analysis. Like, so when we get the FASTQ files from sequencing, you can, I used Metaflan. It used to be human, but now they have Metaflan. So we reanalyzed them using Metaflan. I got the composition and functional data. And but for 16S rRNA, I didn't use Illumina, which is the most common sequencing technique, but I actually used Ion Torrent, which is not very common. So I had some problems analyzing it because the CHIME, I'm sure you know CHIME, right?
Yes.
The software for analyzing 16S rRNA gene sequencing data. So I didn't run CHIME for Ion Torrent data because they sequence various hypervariable regions, V1 to V9. So they have inbuilt software to analyze 16S data. So I use that 16S.
Yeah. So in your case, you use in the 16S rRNA gene, you use from V1 to V9 all covered. You covered all the regions. not a specific region, right? Yeah.
No.
So, and then how about what kind of taxonomy, the database you use to assign the taxa for the microbiome identification?
I can't. It's not in my mind right now. But because we use this inbuilt software in Iron Torrent, and so it's not the one you usually use for Chime, but it actually gives the species level data. I didn't present the species level data, but we actually look at this genus level data because one thing we were not sure whether it's accurate to look at species level data from Iron Torrent. I can actually email you or maybe send you the exact database in Iron Torrent. It's not top of my head right now.
Yes.
Yeah, but it's actually not, you don't follow the same steps as in Chime analysis.
Yeah, right. Yes.
So you just, in Chime, what you do, you can filter, you select the parameters,
Yes.
and what it's actually green. I
Green
remember
genes,
correctly, it's
green. There
green
are like
in
green
database.
genes.
Yeah, green in database. I just remembered. Yeah, it's yeah. That way, I will double check and let you know exactly what it was.
Yeah, thank you. But I will refer to your paper so that I can find the information, right?
Yeah, sure. Yeah,
right.
yeah.
So is there any specific reason go up to the genus level, not going like considering the other taxonomy level?
You mean the species level?
Yeah, like a species going up to the species level, because I saw in your presentation you identify the Prevotella, right? Prevotella genus is kind of like
Yeah, that's yeah.
yeah. So I was curious about whether there is any specific species of the Prevotella. So because in my experience I was able to identify like the Prevotella intermedia is a kind of a species enriched in the gastric cancer cases. So like inflammatory
Oh,
related
that's really
bacteria.
interesting.
Yeah, right. So I was
Yeah,
curious
that's
whether
yeah.
you,
That's,
yeah, whether you were able to identify that kind of the species like related to.
yeah. So yeah. Thanks for asking that question. So as I mentioned before, we didn't actually look at the species level data because we were not 100% sure that species level data from this iron torrent sequencing is accurate to start it. So we didn't look at the species level data. So that's why we went up to genus level data. But your point is very interesting because I don't know whether you remember, but in Billy's data like the recent samples when he was 70 years something, and then we had very high level of Prevotella, but we don't know what species are really, but definitely something related to like disease condition, and he had undergone some surgeries in his gut. So I am not sure what exact surgery is. We were not actually given that information, but it's really interesting because you mentioned that Prevotella intermedia is associated with gastric
Yes,
cancer,
yes,
right?
right.
Yeah,
Very
Thank
interesting
you.
but unfortunately,
observations. Very interesting observations. You two should take notes from each other.
Thank you, Dr. Thilini, for your good answers.
yeah.
Thank you very much, both of you, for your very nice, informative presentations. And I have some few questions to raise, Dr. Thilini, regarding that. So in the first study, you mentioned that you analyzed the fecal and plasma amino acids. What is the method did you use, Thilini? So how did you analyze that? So especially the fecal, the stool samples. So I'm quite interested on that.
So yeah, so we used SPMA, solid phase micro
Okay.
extraction
After
method to
extracting,
extract the
yeah.
amino acids. So are you asking
Yeah,
about amino acids?
amino
So,
acids.
Yes, so we used amino solid phase micro extraction, and then we used sorry, just looking at the fecal and plasma amino acid. No, that's we use solid phase micro extraction for fecal and plasma volatiles, followed by GCMAs. But for fecal and plasma amino acid analysis, we use ultra performance liquid chromatography.
Okay.
I didn't analyze them by myself, but someone else. We had
Okay.
master student, so he
So-
did. But we used UPLC.
Okay. And again, Thilini, that the arginine thing. So you know that this is conditionally essential amino acids. So the preterm babies has got less amount. Is that what you mentioned? No? And extremely preterm, isn't it? Yeah. And compared with the preterm.
Extremely.
What may be the reason? Is there any relationship with the diet or with maternal nutrition? So is there any guess regarding that?
Yeah, so that's a great question, madam. So we actually look. I didn't present those data, but we looked at the diet of these children and analyzed different parameters. We didn't see any change in the diet between these two groups, but from the previous literature, we know that these preterm children have arginine deficiency. They don't. They are lacking one of the genes to produce arginine. So that we presume that this is because of that they have less arginine in their plasma. And when we looked at the microbial functions, and we saw some of the microbial functions related to arginine are increased in these preterm children. So there's definitely a link between plasma arginine level in these preterm children and the microbial function, like microbes who produce this arginine, and some of the microbes which are associated with arginine biosynthesis.
Okay, that means there is a relationship between the microbiome and the arginine production. No, is that? Yeah. Okay. Okay. Thank you. And my
But
last question.
we don't have a direct link. We can't really say
Yeah. But
direct
there
link.
may be associations. So that's
Yeah,
what you
that's right.
predict. Yeah. And then again, my last question regarding that storage of the capsules. So just to know that, how did you do that encapsulations of these fecal matters? What kind of method did you use that? So we are not used to that. That's why just to our knowledge.
Yes. Yeah, so I first of all, when I get the sample stool from the donor, I mix it with saline, and then it's like a slurry, and then filter all these undigested food particles and a lot of different debris, and then take the liquid and then centrifuge it until we get a pellet. We call it bacterial pellet. It's not a pellet actually. It's again like very liquidy, and then we concentrate that, like do a lot of centrifugation, and then concentrate it, and then mix it with saline and glycerine. We have a concentration to make it up to, and then put it in capsules one by one, because it's a liquid. It's it can be leaked, so we actually put it in two different capsules.
Okay.
And once it's we transfer them to -80, and once it's frozen, it's solid. You can't say whether it's stool or saline by just by looking at it. Yeah.
Okay. Thank you, Thilini. So very nice that.
Okay.
So thanks both of you for your very nice presentations, and I'm really proud of you, of both of you, and thank you so much.
Oh, thanks, madam.
Thank you, madam.
Dr. Thilini, I just have just a little clarification in terms of the engraftment. I might have got it wrong, but I think you said that the engraftment lasted for about 26 weeks. Is that so? The capacity of the engraftment?
think there's a delay, no?
Yeah. Not sure if it is from my side. I think
I think she didn't get the question.
Dr. Thilini, I think she's frozen.
Yeah, she's
Yeah.
frozen.
Sorry.
Yes.
Did you get my question? Sorry, we couldn't hear you. I think there's a delay in the connection. Then she's dropped. Oh no, she's here.
I'm sorry, Dr. Dushanti. I didn't hear what you said. I think my
Okay, I
connection
will.
is very
Yeah. There was a connection problem. I was just wondering. I think I heard that
hear
you
me?
said yes. We can hear you.
Sorry, I didn't hear what you said, Dr.
I
Dushanti.
will
Can you
repeat it.
repeat
Yes.
your question?
I will repeat it.
Sorry,
I was just wondering, I think you said when we do the engraftment that it lasted for about 26 weeks, the effects? Is that?
Yeah.
Yeah. So if we say that we are to continue this particular treatment, so that means we have to do the engraftment like every 26 weeks. Is that something that you are looking for, or?
So this particular project was actually designed to check the microbiome after 6 months. Can you hear me?
Yes, we can.
Oh, okay. Yes, maybe I didn't mention that, but we actually gave 28 capsules to these recipients on 2 consecutive days. So first day 16 and the next day, I think 12.
Okay.
So after that we followed them up for 6 months, and we still see change, which was lasting for that long. But we don't know whether, because we finished that study after 6 months, so we don't know whether we should do another transplantation.
Okay. So that's why I think we need long-term studies looking at how these the effects would change from time to time and what their influence on the overall microbiome and health. So yeah, got it. Thank you.
Thank you, Dr.
No
Tushan
worries.
and Dr. Malkanti for your valuable questions. So from the audience, if you have any questions, you guys can raise your hand, or else you can direct your questions in the chat box. So we are waiting to get your questions. Yes, for the very first question, we have Shihari Jayasinghe. Yes, Shihari.
Yeah, thank you, Kosala, for giving me the opportunity to raise my question. Good morning to all of you. I have 2 questions actually. So both of my questions goes to Dr. Thilini. Very good morning to you, ma'am. First and foremost, thank you so much for your very informative and interesting presentation. And it made me go through your PhD thesis. So I just went through your PhD thesis at a glance. So I found that something, a subtopic in your thesis, irritable bowel syndrome. So we've been taught about the irritable bowel syndrome. I'm just interested about how it can make an influence on the population of human gut microbiota, whether it's negative or positive. And my second question is, among the factors influencing the development of microflora in the human gut, which factor is most influential? Thank you.
Okay, so I think I didn't hear the first part of the question, but I think you asked in general what the relation, like what type of factors affect on the irritable bowel syndrome. Is that right?
Ma'am, I just want to know how it affects the gut microbiota population, because we've been taught about irritable bowel syndrome. It's not harmful to our body. So I just want to know about what it does to the microbial population.
Yeah, so yeah, I'm actually not researching about irritable bowel syndrome, but from the literature, it says that yes, there are some differences between irritable bowel syndrome patients and healthy controls. I don't know what exact species, maybe Dr. Madhawa can help me, but I don't know what exact species are actually highly abundant in irritable bowel syndrome. But as a whole, the diseases linked with the digestive tract actually change your bowel a lot. Like your microbiome is different when you have a disease condition in the gut. So it's same with taking probiotics and prebiotics, antibiotics. Yes, but I am not sure what exact species are actually associated with irritable bowel syndrome.
Yes, madam. And my second question to you is, among the factors influencing the development of microbiota, which one is most influencing on the population? I mean, in your thesis, you have mentioned that the environment, the genetics, the breastfeeding and formula feeding, so many factors. So I want to know about which one affects more.
Yeah, that's a great question. So, yeah, so as I mentioned before, there are so many factors which shape up our microbiome at different stages of the life. And according to, I mean, in my research, again, I didn't look at which factor actually impacts on the microbiome most. When we look at the diet, we didn't see any change, in my cohort, in different cohorts of the projects. But from the literature, it says that 55% of the change in microbiome is coming from the diet. So diet is the major factor which is actually changing your diet for good or bad. Yeah, so to answer your question, diet is the most influencing factor which actually changing the microbiome in humans.
Okay, thank you very much, madam. And Dr. Madhawa, thank you very much for your presentation
Thank you.
too.
Thank you, Dr. Thilini, for clearing out the question, and thank you, Shehari, for raising the questions as well. So I'm handing over to Niru. Yes, Niru.
Thank you, Kosala. So thank you for this session, and thank you, Sahari, for asking the questions. So is there anyone who would like to ask the question? So we have 10 more minutes to go. Yeah. So now you can ask your questions to clarify your doubts.
Good morning, everyone. I'm Senuri here. So thank you again, Dr. Madhawa and Dr. Thilini, for this informative presentation and explanations. And my question goes to Dr. Madhawa. Doctor, you said that fruit consumption is associated with a reduced risk of cancer. So how much amount is normally adequate to take daily to get this benefit?
Thank you for your question. So so I didn't understand the clearly the question about how much you mean the intake amount, like a gram.
Yeah, the intake amount.
Yeah. So in this for this question, I can answer like it's very difficult to give a exact value. So for considering a single food item or the food group, or but certain recommendation allowances are, so those you can refer to have idea about that kind of intake based on the previous recommendations, dietary guidelines. However, when it comes to the dietary patterns, so the patterns address the complexity nature of the diet. So in that case, we are not going to consider a single nutrient or a food group at one time as a single way. So we consider an overall picture of the diets. So it represents the kind of the overall picture of the diet, and that's how I can answer for that one. So specifically, I cannot give a specific value or a kind of the answer for that question. But one thing is you can refer to the dietary recommended RDA values, so those kind of information to get an understanding about for the the for the prevention of cancers, what kind of food should we have to use. But so those, and when it comes to those RDA and those information, so those are derived for the general population, I guess, I think. So, so in this case, when it comes to the disease association, so we have to consider about the previous research, like based on meta-analysis, so and cohort study and case control study findings. So they are pooled effect. We have to consider about have an idea about what kind of food groups or the the proportion or the portion or item that we have to consume for the disease risk reduction. Did you get the answer for your question?
Yeah, it's clear, Doctor. Thank you.
Thank you, Dr. Madhawa, for answering the questions, and thank you, Senori, for asking your questions. So we have 5 more minutes. So is there anyone who would like to answer the question, ask the questions? Yes, you can go forward.
Good morning, all of you. I'm Chatumalika. Thank you very much, both Madam and Sir, for delivering a very valuable webinar today. I would like to know, Sir, is there any effect of frequently coffee consumption and the gastric cancer? Is any link between coffee and this gastric cancer? Can you a little bit explain it, Sir?
Yes, that is also interesting question. Actually, recently I was able to work the coffee consumption and the colorectal cancer risk association, and in that case we were able to identify a very significant reducing risk of the high consumption of coffee and the colorectal cancer. However, when it comes to the gastric cancer, in my experience, I didn't have work with that, but according to the bioactive compounds present in the coffee, specifically like caffeine and certain other the compounds, they can have the anti-cancer effect or anti-inflammatory effects to reduce the risk of gastric cancer maybe. So one thing that I can tell you that you can refer to certain papers related to the cohort or the case control studies, findings about the association between coffee and gastric cancer. For the colorectal cancer, I can exactly tell you that it has the significant inverse relationship for the colorectal cancer risk.
Okay, sir. Thank you.
Thank you, Dr. Madhawa, for the answering. And we have one more questions from the so is only the dietary pattern or mentality caused to the gastric, which is mostly affected the cancer risk as a percentage?
Yeah, so the...
Excuse me, am I audible to you?
Yeah, so for the question, so there might be interaction effect with the patterns or the diet and the mentality for the gastric cancer. So these are like a psychological epidemiology related questions. So in my experience, I cannot exactly answer for that one. But the dietary patterns has like a healthy, or it depends on the healthy pattern or unhealthy pattern. So they are the specifically healthy. When it comes to healthy patterns, it may related with the reducing risk of the cancer, and for the unhealthy pattern, so those patterns are definitely associated with increasing risk of the cancer. So for the psychological point of view, I'm sorry, I cannot give any the answer for that. And yeah.
Okay, sir, thank you so much. So I have found that there are two more other one who raised the hand button. So you can ask your question directly. Hashini?
I think there are two more questions from the audience, but unfortunately, I'm not sure if they are with us due to internet connection issues. So until they come back, Dr. Madhawa, just a little clarification. So when you are feeding data to GGM model, do you feed physical activity data as well to get the patterns?
Yeah, so for the GGM analysis, I did not consider the physical activity data as input variable. I only considered
The
the
diet.
food diet only. But when it, as I mentioned in my presentation, the reduced rank regression. So if you need to consider about the considering response variable, so the disease specific nutrient or disease nutrient ratios or certain other related variables could be considered for the in that analysis, but in the reduced rank regression. But for the GGM, I only considered the food group variables as the input variable.
Okay, so does that mean the GGM is only sensitive to dietary data, or can we model other types of data that we collect?
Yeah, that is a great question. So I think we can use the possible confounding variables like the physical activity data and the information to model and then identify the kind of like in holistic manner to identify the network. Yeah, that's a very great question.
Yeah, I think that would be, I mean, it will probably give us a more holistic look. I mean, diet is already comprehensive, that we understand that. But again, I think if we can look at different GTM models feeding different variables, so we can, I think it will probably complicate the graphics, but I think that would be a really nice way to, move forward, looking at the comprehensive nature of the diet, the physical activity, the diseases, the medication, because all of them work as one rather than they work separately. So I think this model is really interesting. I think I heard it one time, but I never had a chance to go through it. And it was really great opportunity for me to get to know that from you. So if I have more questions, I will contact you. I'm very interested in that model.
Yeah, definitely. Feel free to contact me.
And I will read your papers.
Yes. I was able to publish this paper in Cancer's Journal, and yeah, so you can refer. And if you have any question, please email me.
Thank you very
directly.
much.
Yes.
I think we have a question now from the audience.
Yes, madam. Thank you, madam, for asking the question. And we have Hashini here. Yes, Hashini, you can direct your question.
Hello, I'm Hashini here. Good morning to everyone, and I'm very thankful for very interesting presentations from Dr. Madhawa and Dr. Thilini. My questions goes to Dr. Thilini, and I would like to know does it how long does it take to regain our microbiome in conditions like infections and diarrhea like conditions?
Are you asking from me?
Yes, madam.
Okay, sorry, I was not sure whether it was directed to me. So that's a very hard to answer that question, first of all. I'm not sure exactly how long it will take, to restore the microbiome. Are you asking about any specific disease or in general?
Conditions like infections and diarrhea like conditions normally.
Sorry, say it again, please.
In conditions like infections.
Yeah, so definitely, when you have an infectious disease, like your microbiome is changing, and it's actually a short-term change. So you can restore the microbiome with a diet, for example. I don't know how long it says, how long it exactly takes, but it says short term. But when you are on antibiotics for so long, your gut microbiome can be changed sometimes, for a long time. And it is hard to restore that microbiome unless you follow a certain diet, like, with fermented food, and sometimes prebiotics and probiotics. So it will take a while to restore. But infectious disease, if I'm not sure what exact infectious disease it is, but if it's a normal kind of flu, for example, your microbiome is changed. I'm not talking about when you are on antibiotics or some antiviral effect drugs, but in the normal scenario, if you are on a certain disease, your microbiome can change short term, but you can restore with diet within pretty much short term, I would say.
Okay, madam. Thank you very much.
Since we are running out of time, we can take only one question. So I can give chance to Devni Nisanka. So in the chat box, I think this question will goes to Dr. Jayani. Dr. Thilini, in celiac disease, will elimination of gluten in diet leads to complete restoration of gut microbiota or partial? In addition, can lactose intolerance be corrected once again? Yes, Dr. Thilini.
Oh, thanks for the question, but this is not my area of expert. Like, different diseases and gut microbiota, especially gluten. I am not sure. Maybe you have to look it up on the literature. How the elimination of gluten will lead to complete restoration of gut microbiota or partial. I'm sorry, I can't answer that question. The second one, can lactose intolerance be corrected once again? I don't know that answer as well. I'm sorry. But there are some species, like H. pylori, for example, associated with lactose intolerance. And if you take and it comes from the lactose intolerance is coming from its genetic one as well. So, but maybe part of that can be corrected with correct use of fermented foods, for example. I mean that fermented dairy foods like yogurt. You can correct that. I don't think you can fully correct that, but I might be wrong. Maybe Madhawa can answer that question.
I'm also not much sure about the question, so sorry for that.
Okay, Dr. Thilini and Dr. Madhawa for answering the questions. Very thank you. So it's Shehan, your turn to deliver the word of thanks.
Very good morning, everyone. It's great pleasure for me to deliver the vote of thanks on this occasion. First and foremost, I thank our special speakers, Dr. Thilini Jayasinghe Adikari and Dr. Madhawa Gunathilaka, who despite their busy schedules have found time to this occasion. And I was also express my heartfelt thanks to Dr. Kumari Malkanthi Ratnayaka, head of the department, for the valuable contribution, guidance, and encouragement in all our efforts. We are very grateful to our madam, Dr. Tushanti Pereira, especially for contacting the special guest speakers today, and also for the guidance, encouragement, and support given to us always being behind our back. I would like to express my sincere gratitude to all our lecturers of the Department of Applied Nutrition for the guidance and support given to us to make this event successful. I also would like to thank members of the Nutrition Society of the Department of Applied Nutrition for the support and contribution given. Last but not the least, I thank all the participants and all who were with us in making this event a resounding success. Once again, thank you for your attention.
Thank you, Shehan, and thank you, Nutrition Society, for all your support and contribution to make this a success. And thank you very much, Dr. Thilini and Dr. Madhawa. As I said, amid their busy schedules, they agreed to participate today, and we have been trying to arrange this webinar for a couple of months now. So they have been always responding to me. So thank you very much for making my life easy and doing all the preparations necessary to arrange this webinar today. So I think this has been a really, really great opportunity for our students. So we will be uploading webinar recording to our Nutrition Society website. So please follow our Facebook page for new updates as well as the Nutrition Society website. So if you have any further questions or concerns, or if you would like to read more about these new methodologies that have been introduced today, our presenters talked about their publication. So you can definitely go and check them out, read them. That would be a really great learning point for you as well. So if there are any questions or concerns, you can direct them to me, and if time permits, I will pass them down to Dr. Madhawa and Dr. Thilini. With their time how much time permits to them, they will probably get back to us. In the meantime, you can always follow us, our Facebook page and our website, and go through the webinar again if this is something that you are really interested in, and if you want to know more details and information. So we are right on time. It's 11:02. So we will be winding up today's webinar session. I'm sure Dr. Madhawa and Dr. Thilini will have a lot lined up this Sunday. So thank you very much again for joining with us and spending 2 hours with us. So we are very grateful. Thank you.
Well,
Hoping to continue this, this dialogue in future as well. So don't be strangers. Please write to us. Please stay in touch with us.
Thank you. Thank you, everyone.
Thank you so much.
Accepting our invitation, and I just met the 2 of you, so I need. So that's very great. So to see you. And
thank you, madam.
Thank you, madam.
Thank you, and joining through all the way.
Yes. So thank you all the participants. We had about 140 participants, so that's a great turnout.
Yeah.
So thank you to everyone. Have a good Sunday. Enjoy your rest of your weekend.
Thank you, and have a good weekend. Bye.
Bye.
Bye. Bye.
Bye, madam.